Vaping is a trend that has increased exponentially in the last few years, especially among the vulnerable population of high school students. In 2021 alone, an estimated 5.2 million high school students and 1.34 million middle school students reported ever using a tobacco product, the majority using e-cigarettes or vapes. Why has this increase occurred so rapidly? There are many reasons, but to name a few, the misperception of safety surrounding e-cigarettes since they do not expose your lungs to tar that tobacco products would, but many users would consider them less harmful, or maybe they think of them as just water vapor or juice, but not nicotine. They are also considered less taboo. As prevalence has increased, it has become more socially acceptable and even expected that people use vaping products. Especially in adolescent populations, peer pressure and high availability contributes to increased experimentation and high addiction liability if adolescents are exposed during this developmental window to nicotine.
I was myself really shocked when I first saw the stats, how many high school students are using these products. The reason that we are specifically focusing on teens is that teenagers are uniquely vulnerable because their brains are still in rapid growth, synaptic pruning, and remodeling of reward pathways. Nicotine hijacks this process, overstimulating dopamine circuits, rewiring habit forming networks, and weakening attention and emotional regulation in ways that are harder to reverse than in adults. Focusing on vulnerable populations is another objective in this podcast. And we're going to focus on the paper Nicotine on the Developing Brain by Emily Castro, Frances Leslie, and Shahrzad Lotfipour. Aside from teenagers, pregnant individuals are one of the most vulnerable populations. Since quitting nicotine at that age is incredibly difficult, the evidence shows that the nicotine alone can disrupt fetal brain development, making it the main contributor to the harm in babies developing central nervous system. In this paper, we're gonna show that nicotine experimentation in teens is a major contributor to altered reward pathways, which is also known as gateway effect, where teens are become more prone to experimenting drugs of abuse in later life. And as I said, nicotine itself is the major contributor to the developmental harm of the fetus, not other harmful products in vapes. We will introduce these effects throughout this episode, but the key takeaway is that the introduction of nicotine is detrimental to developing reward pathways if introduced at any stage of development, regardless of administration method.
Welcome to the Translational Addiction Podcast, where we decode the innovative research from the Lotfipour Lab at UC Irvine, dedicated to translational addiction studies and developing effective treatments. I'm your host, Michaela Somorton, and I'm a fourth-year neurobiology major at UC Irvine. And today with me, I have another co-host. I'll let him introduce himself right now.
Hello, I'm Ardavan Merhazma. Very happy to be here. I just joined the Lotfipour Lab. I'm a third-year bio major at UCI, and it's very exciting for me since this is my first podcast. I hope you really enjoy it. And I just want to jump into our interesting points. As we mentioned before, nicotine itself is responsible for developmental harm to a fetus, but it is difficult to truly identify the extent of exposure to nicotine in a developing fetus because of factors such as mode of intake and frequency of use that confound clinical studies. It is a common misconception that nicotine replacement therapies, more commonly known as vapes and e-cigarettes, are a safe alternative to smoking tobacco products during pregnancy. But nicotine can cross the placental barrier during fetal development, which alters neural pathways and other developmental processes that we will discuss today. We will elaborate on our earlier discussion regarding nicotine as a gateway drug by reviewing the paper Nicotine on the Developing Brain. We will outline clinical and preclinical studies that provide insight to reasons why nicotine specifically is the mechanism by which developmental issues occur in utero, and nicotine use during adolescence may alter reward pathways, leading individuals of using more addictive drugs such as cocaine later in life, also known as the gateway effect. Just a side note, our second podcast, Neuroglia and Substance Use Disorders, did mention these concepts. I really encourage you to listen to that too. but was more focused on recent findings in the ways microglia and astrocytes mediate addiction. Whereas this podcast will serve to outline nicotine as a gateway drug, as well as the brain regions affected both in utero and during adolescence, which are two critical developmental windows in humans. I want to start from prenatal nicotine exposure, which is during pregnancy. In that period, we have, of course, our early brain development, and it is very important to little expand on this. So, our brains develop and organize themselves from the inside out, from inside to outside. In mammals, generally, in both humans and rodents, deep neural layers are formed first. So we may study the effects of nicotine on a developing fetus both clinically and preclinically. As we mentioned, nicotine can cross the placental barrier and interact with nicotinic acetylcholine receptors, or NACHRs, throughout the whole fetus, but especially in fetal brain. Normally, endogenous ACH should bind and activate NACHRs, or nicotinic acetylcholine receptors, but nicotine acts as an agonist, and it's not endogenous, it's exogenous, it's coming from outside, meaning that it binds to NACHRs and activates them. If you want to learn more about NACHR activation by nicotine, again, check out our second podcast, Neuroglia and Substance Use Disorders.
This episode will review each brain region in the review article, beginning with the most relevant reward circuits that are indicated in the gateway effect during adolescence, before moving to more primal regions such as the pons and medulla, which are indicated in sudden infant death syndrome after prenatal nicotine exposure. So we'll begin with the ventral tegmental area and nucleus accumbens. The mesolimbic pathway is the first brain circuit we will discuss, and this is because it's involved in reward and motivation associated with development and reinforcement of drug addiction. The primary connection involved in this circuit is between the ventral tegmental area, or VTA, and nucleus accumbens, or NAC. And the primary neurotransmitter that is released is dopamine. So the VTA and NAC are therefore major sites of nicotine-induced dopamine release. And the mesolimbic pathway is also indicated in all reward paths that may be hijacked by drugs of abuse. So in clinical studies, these have established that nicotine exposure during adolescence but not adulthood is associated with subsequent increases in substance use later in life, which is essentially the gateway effect. In models, treatment in early adolescent rats with low doses of nicotine, equivalent to 1 to 2 cigarettes per day over 4 days, intended to model smoking initiation, results in significant increases in self-administration of cocaine, alcohol, fentanyl, and methamphetamines. This phenomenon is not observed in adults in most cases. Nicotine pretreatment in animal models has shown increases in microglial markers, D2 receptors, G protein coupling, and CFOS mRNA expression as well in adolescents, but not in adults. These patterns are associated with increased addiction liability. Conversely, in adults, nicotine actually has the opposite effect of downregulating microglial markers and D2 receptors, and this concept is a major focus of our second podcast, Neuroglia and Substance Use Disorders. So for prenatal and perinatal, the effects of nicotine exposure on the VTA and NAC are not discussed in the context of these developmental windows, but when we discuss other brain regions, prenatal will be the first 2 trimesters of gestation in humans, and perinatal refers to the 3rd trimester through the first couple of weeks after birth, so just the time surrounding birth, which will be relevant to other areas of the brain during development.
As we mentioned, we have this inside-out developmental model in our brain. So when we talk about cortices, especially prefrontal cortex, we usually see the effects in adolescence because cortex develops later in life. But let's start with sensory cortices. So in adolescents and teens, Clinical studies have revealed that children with mothers who smoked during pregnancy have difficulty processing auditory tasks in a sex-dependent manner, which means girls and boys have different symptoms. Girls usually have impaired somatosensory and visual processing abilities, while boys usually show impaired ability to perform auditory tasks. During early postnatal development in rats, which corresponds to the third trimester in humans, topographical projections from thalamus to the cortex are refined by inputs from the external environment. During a critical developmental period, transient alpha 7 nicotinic acetylcholine receptor expression in the sensory cortex and thalamus facilitates NMDA receptor mediated thalamocortical circuit formation. If I want to simplify, this circuit between thalamus and cortex is being formed. And during this time, if nicotine is present, what happens is that it disrupts this process by altering NR2B mRNA levels. So you won't have that precise point to point projection topographical maps in your cortex. Nicotine treatments during this period eliminates normal NACHR signaling in the adult auditory cortex and impairs performance on auditory cue-based avoidance tasks without affecting basic auditory perception. What it means is that your normal hearing is okay. You can hear faint noises, but higher cognitive processing in your brain is impaired. Collectively, these findings show that nicotine exposure during critical period of sensory cortex development impairs cognitive tasks that are dependent on sensory cues but does not alter normal sensory function itself. So it's a bit harder to assess this as a clinician. Maybe the child or the teenager shows normal function, processing very simple sensory information, but they have problems in higher order cognitive processing. Now let's get into prefrontal cortex, which is my favorite region that is being investigated in this paper since it is correlated to the maturation of executive control, emotional regulation, higher order cognitive abilities of humans. So in adolescence, this area is undergoing very important changes. What happens is that in teens, increasing cognitive capacity occurs alongside a decrease in cortical gray matter thickness, reflecting experience-dependent strengthening of active synapses and the natural pruning of weaker connections. During late adolescence, there's a decrease in connectivity between subcortical regions and increase in top-down regulatory control by the medial prefrontal cortex, resulting in reduced emotional reactivity. In other words, cortex has a more effective control on those subcortical regions, so we can regulate our emotions easier. Smoking in early adolescence increases impulsivity and inattention due to alterations in PFC function. Again, PFC plays a very important role in human intelligence, and early onset smoking leads to long-lasting decreases in task-related attention behavior, again, inhibitory control behavior, and prefrontal attention network function. You have impulsivity and lack of attention. Teen e-cig use is associated with lower academic achievement, and there are no significant differences between the effects of e-cigs and combustible cigarettes on school performance, highlighting the key role of nicotine itself. So there's no difference between vapes and cigarettes in lowering academic achievements. In animal studies, adolescent nicotine exposure, but not adult, negatively affects cognitive performance on PFC-dependent tasks, producing increased impulsivity and reduced attention in adulthood. So these effects are prolonged. The underlying mechanism involves lasting synaptic alterations in the PFC. Adolescent rats exposed to nicotine for 10 consecutive days have been shown to reduce their inhibitory presynaptic mGlu-R2 protein expression and function. And this protein is crucial in synaptic plasticity. So when you have a reduced amount of this protein, you have impaired synaptic plasticity in your brain. All of these neurochemical changes are accompanied by neural hyperactivity, reduced D1 receptor expression, which is a dopamine receptor, and increased phosphorylation of ERK1/2 in the PFC. And overall effect is alterations linked to the mood and anxiety-like behaviors. This pattern is consistent with clinical findings showing a bidirectional association between depression and adolescent nicotine use. If you are a teen and you smoke, you have a higher chance of depression later in life. It has been shown that prenatal nicotine exposure in this period of time increases dopamine levels in the prefrontal cortex, striatal D3 receptor binding, and D2 receptor functional coupling in the ventral striatum and pallidum. It has been also showed that in the prenatal period, nicotine exposure throughout gestation alters best firing of dopamine neurons, increasing the heterogeneity and complexity of neurons in subregions of the ventral tegmental area, and is also associated with a decrease in the nicotine-induced striatal and cortical dopamine release.
So now we'll be moving into the midbrain and hindbrain, so including the hippocampus, pons, and medulla. So the hippocampus is associated with learning, memory, and spatial navigation. It continues to mature throughout adolescence in humans, with significant development occurring in the first 2 years of life and later maturation between the ages of 9 to 11. So, animals can be used to model varying windows of development in humans. For example, rodent postnatal week 2 is comparable to the third trimester in humans. Preclinical studies have found that the hippocampal cholinergic system undergoes transient upregulation of nicotinic acetylcholine receptors during regular critical developmental events in this time period. And the immature hippocampus is characterized by giant depolarizations from the synergistic interaction of glutamatergic and GABAergic neurons. So GABA normally acts as an inhibitory neuron, but in this stage of development, GABA is actually excitatory because of the immature chloride gradient. And the introduction of nicotine increases these giant depolarizations via the activation of GABAergic neurons, which are again excitatory during this period. So nicotine and endogenous acetylcholine increase synaptic efficacy in early postnatal glutamatergic synapses. And in later postnatal development, alpha-7 nicotinic acetylcholine receptors are shown to control the timing of GABAergic conversion from excitatory to inhibitory signaling. And chronic early neonatal nicotine exposure in rats increases neurotrophic tone during this period of hippocampal development. So, in adolescence, the effects of nicotine exposure include long-term impairment to hippocampal dependent spatial memory, and the hippocampus is interesting because you actually have place cells within the hippocampus that you can actually track maybe a mouse running through a maze, and as the mouse goes to different areas of the maze, you can track place cells, and a certain area of the maze will correspond to certain place cells in the brain. And so nicotine is also seen to alter long-term potentiation as well, because the hippocampus is highly involved in learning and memory, and long-term potentiation is considered a form of memory establishment because it's the activation of neurons over a long period of time, which increases synaptic connections or increases plasticity. Nicotine is also seen to alter long-term potentiation. Loss of normal alpha-2 nicotinic acetylcholine receptor function is observed, and impaired NMDA receptor dependent long-term depression is observed in adolescence, and these all are contributing to the decrease in learning and memory ability. So the pons develop in early gestation and reside in the brainstem. Pons control unconscious processes, including sleep and respiration. The pedunculopontine tegmental nucleus, or PPTG, within the pons is critical for sleep, arousal, attentional, and motivational stimuli. So in prenatal models, prenatal nicotine exposure has an overall excitatory effect on the pons. And the offspring of pregnant rats that are exposed to nicotine or cigarette smoke during pregnancy change the cholinergic neurons of the PPTG. alterations of the PPTG are heavily associated with the sudden infant death syndrome, or SIDS. So SIDS is the sudden and unexplained death of an infant under 1 year old. And although the exact cause is unknown, sleeping on the stomach or side, soft bedding, overheating, and maternal smoking during pregnancy or breastfeeding are known risk factors. Neurally, this is possibly related to the PPTG involvement in REM sleep and control of respiration. Human studies that evaluate brain tissue following SIDS in infants exposed to nicotine through maternal smoking show a decrease in nicotinic acetylcholine receptor binding in the nucleus pontus oralis, which is another area of the pons, and this area is a critical component of the cholinergic ascending arousal system. The medulla oblongata is vital for regulating involuntary functions such as cardiovascular control, respiratory control, and digestive control. Organisms seek to achieve homeostasis, or a stable internal environment, and the medulla is involved in the regulation of these. Prenatally and in clinical trials, altered nicotinic acetylcholine receptor binding is seen in 3 medullary sites closer to the front of the medulla with serotonergic or 5-HT neurons. It is known that nicotine impacts serotonergic function. For example, monkeys exposed to nicotine prenatally show significant serotonergic hyperinnervation of the brainstem, which is a hallmark of SIDS. In prenatal mice study, nicotine exposure decreases the firing rate of the raphe obscurus neurons in neonates and increases the expression of serotonin autoreceptors. The raphe is associated with the production and release of serotonin, and that's a neurotransmitter known to modulate mood, sleep, appetite, and pain perception. And increased expression of autoreceptors indicates an enhanced negative feedback mechanism, leading to a decrease in the synthesis and release of serotonin.
For those who are interested in genetics, I think this is a very interesting part. It was one of my favorite parts in this podcast as well. And we want to talk about epigenetics. I think you are aware of how smoking can cause mutations in your DNA, and basically your gene sequence will be different. Here, what we're going to talk about is epigenetics, which means that your sequence of bases in DNA is okay. So basically, you'll have the same sequence of nucleotides. But the thing is, you have these chemical sticky notes, which means that on the same sequence, you can have some chemical alterations, which are reversible, but they can also be passed to the next generation. So nicotine and other drugs of abuse can trigger such modifications. This is very important because all these changes can decrease or completely silence some of your genes. So you will have less output or less proteins created from that same gene sequence, which can have very serious developmental outcomes. So when we talk about prenatal period, this period is characterized by high epigenomic plasticity, crucial for brain development. So smoking mothers have newborns with differentially methylated CpG sites in neurodevelopmental genes. CpG islands are found near promoters and can alter expression of genes. So when you have these methylated CpG sites, they generally suppress gene expression. So you have this widespread suppression of gene expression in neurodevelopmental genes. And these changes appear at birth, they persist into adolescence, and they are linked to schizophrenia and other neuropsychiatric disorders. Exposure to nicotine in utero can increase the DNA methylation in fetal cortex, and this is unfortunately linked to decreased neuronal count. This is very shocking. Grandmaternal smoking is associated with higher asthma and autism risk in grandchildren. In mice, gestational nicotine alters DNA methylation around PPAR gamma, a gene promoter. This gene is very important in developing the lung tissue. And when you have alterations in expressing this gene, you will have an impaired lung differentiation. Interestingly, this persists to generation 3. So not only your grandchildren, but a generation after them. let's go into the perinatal period, which is the postnatal period. So the exposure at this time remodels sperm DNA methylation. Like, again, repeat DNA methylation can suppress gene expression, transmitting epigenetic changes across generations. So generations 1 and 2 show increased nicotine intake, hyperactivity, and risk taking, alongside reduced nicotinic receptor and dopamine transporter function. Altered BDNF is very important in brain health and stress tolerance. Brain-derived neurotrophic factor, which is important in creating new synapses, and disrupted HPA axis. So HPA axis controls the excretion of cortisol, our stress hormone. So this disrupted HPA axis activity impairs reward systems and reward pathways and stress pathways. So you have lower stress tolerance. And adolescent nicotine exposure can lead to cognitive deficits in the offspring. And nicotine and stress together reduce nicotine sensitization, altering brain's reward response. So you may ask, what's the problem with reduced nicotine sensitization? Like, at least I thought it couldn't be a bad thing at the beginning. But the thing is, when you have this reduced sensitization to nicotine, it means that something is wrong with your reward pathways. And this can lead to anhedonia, which is inability to feel joy, and you will be just depressed, and it makes you more vulnerable to addiction. This change persists across generations, especially in F3 females, showing that adolescent exposure can epigenetically pre-program vulnerability to abnormal stress responses and future addictive or mood disorders. Paternal nicotine exposure, preconception, can have long-term heritable epigenetic alterations. It can persist for 3 generations. This challenges the genetic reset concept, and nicotine's influence, of course, spans across generations.
Developmental periods like adolescence and gestation are marked by heightened brain plasticity, making them especially vulnerable to the adverse effects of nicotine. While this episode is not a comprehensive review, omitting areas like the medial habenula, it does emphasize that nicotine alone, regardless of delivery method, can significantly disrupt brain development and behavior. The perceived safety of non-combustible products like e-cigarettes has led to increased use among these populations, compounding risks when prenatal exposure is followed by adolescent use. Emerging research suggests that prenatal nicotine-induced changes may amplify the effects of later exposure, with long-term consequences for reward-related behaviors. Epigenetic modifications and transgenerational transmission further underscore the lasting impact of nicotine on brain circuitry. Advancing our understanding of these mechanisms is essential for public education and policy reform aimed at reducing exposure and correcting misconceptions. In 2021, about 18% of high school students reported current e-cigarette use, with 5% using them daily, a troubling rise in habitual vaping. Over 84% of youth users preferred flavored products, which increase addiction risk and appeal. These trends reflect the normalization of vaping despite its known risks to brain development. If you're interested in learning more about the way nicotine alters the developing brain, follow us on Instagram at Translational Addiction Pod for updates, and check out the infographics and papers on the Lotfipor Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipor Lab at UC Irvine, exploring how to understand addiction and develop